Hello, this is Roxana Sanchez Ingunza. I am the owner and manager of RSI Poultry Veterinary Consulting LLC. And in this opportunity, I'm bringing to you the results of a collaborative study with Dr. Martha Pulido from Mississippi State University and Dr. Jean Gard from the USDA ARS in Athens, Georgia. And it is the characterization at the genomic level of Salmonella enteritidis isolated from poultry samples collected by the FSIS in Mississippi between the years 2016 and 2020. The idea to conduct this study originated from information we received during the USDA FSIS virtual public meeting that happened last year in September, where they showed this data that comes from the National Outbreak Reporting System and available at the CDC website, where it indicates that in the early 2000s, more of the Salmonella enteritidis outbreaks in humans were due to eggs. This changed, and in the late years of the 2000s, we observed that more of the Salmonella enteritidis outbreaks in humans are now linked to chickens. In order to try to understand the situation, and as a first step, we decided to characterize at the genetic level Salmonella enteritidis circulating in poultry, taking advantage of the whole genome sequencing data that is available from the FSIS on the isolates they recover from the regulated poultry processing plants. We focus on Mississippi, and we produce information that can be used to guide Salmonella control interventions in poultry. The whole genome sequencing data is available at the National Center for Biotechnology Information website, the NCBI website, and it comes from different sources. It may come from FDA, from the CDC, and from the FSIS. These agencies started using whole genome sequencing in the beginning only for outbreak investigations. The FSIS started using whole genome sequencing in 2016 also for the isolates that they recovered during routine sampling of the regulated poultry establishments. The NCBI data is open access and available to everyone. We downloaded a total of 97 Salmonella enteritidis genomic information from isolates recovered from poultry samples collected by the FSIS in Mississippi between the years 2016 and 2020, and we included a representative number of isolates within each of the sources categories that the FSIS shows at the NCBI website: chicken carcass, young chicken, comminuted, and raw intact chicken. We use 3 Salmonella enteritidis genomic informations as references, including a PT13A isolate from the United States, a PT4 isolate from England, and an isolate from a chicken product from Shanghai, China, isolated in 2013. The data downloaded from the NCBI website consisted on raw reads, assembled contigs, and closed genomes in the case of the references. We use the data to build single nucleotide polymorphisms or SNP phylogenetic trees using the Genius Prime software. We also review SNPs that result in premature stop codons. If a gene is present and it contains a premature stop codon, the gene is not functional. We also look for antimicrobial resistance. We also look for plasmids using these searching tools that are available at the Center for Genomic Epidemiology. And we also accessed the rapid annotation using subsystem technology, or RAST, to annotate the genes, looking for genes that participate in fimbrial biogenesis, virulence, stress response, among other functions of the Salmonella cell. This is the single nucleotide polymorphism phylogenetic tree in a circular representation, showing the Mississippi isolates and color code based on the year of isolation and compared to the reference strains. The first conclusion from this analysis is that the Mississippi isolates are all genetically very closely related and more similar to the phage type 13A isolate from the US. The second conclusion is that the isolates from Mississippi clustered in 2 main big clays. The clay number 1 that can be subdivided in subclay 1A and subclay 1B, and clade number 2 that contains subclade 2A and subclade 2B. We also observed two isolates, one from 2019 and one from 2020, that were clustering away from these two main clades, and we denominated this group as clade number 3. The data was not intended to be used to study the evolution of the isolate over the years. However, we observed a tendency of having more isolates from the years 2018, 2019, and 2020 located in this clade number 1. We identified 5 types of plasmids: the incompatibility F1b and the incompatibility F2 plasmids were found in most of the isolates and in each of the subclades and clades detected. And the incompatibility X1 plasmid was only found in the isolates in this subclade 1A. And one of the isolates in the subclade 2B also contained 2 additional plasmids called an incompatibility X4 plasmids. The incompatibility X1 plasmid that characterizes the isolates in the subclade 1A had 2 important findings. The first one is there were several genes that were missing. They were not found. And among them were genes associated to resistance to beta-lactams, resistance to quinolones, and also in the formation of fimbria. Genes that were present were associated to plasmid conjugation, DNA replication, stabilization of the plasmid, but also genes that belong to the type 4 fimbria. Type 4 fimbria is a specific type of fimbria that anchors in both the inner and outer membrane of the cell membrane of the Salmonella cell. And it is used as a channel to move or mobilize proteins or DNA across the cell membrane. It is also associated to pili and can be assembled or retracted according to the cell functions. It is very important in horizontal gene transfer. It is important in virulence, and it is important also in the survivability of Salmonella cells inside of the host cell. It's been suggested also to be important in the formation of biofilm, and also it's been suggested as a candidate for vaccine development. On this slide in the table, we are showing presence or absence of certain genes in the plasmids. The dark color squares represent presence, the light color squares represent absence. Every row represents an isolate, and a group of isolates from each of the subclades or clades is shown in the table. Based on the 3 main plasmids that were detected, we can classify the isolates from Mississippi in 4 types. The first one is those isolates that contain all the plasmids. We have also isolates that contain only one type of plasmids, the incompatibility X1 plasmid or the incompatibility F plasmids. And then we have another type of isolates that do not contain any type of plasmid. The ACTX and the PIL-X genes are linked to the presence of the incompatibility X1 plasmid. The PET fimbria genes and the SPV genes are linked to the presence of the incompatibility F plasmids. An important observation here is also, for example, this gene, the SPVD gene, even though the plasmid is present, the gene might not be present. Losing genes and also genes that are flanked by mobile elements, like in the case of these SPV genes, suggests maybe the occurrence of recombination events between the genetic material of the plasmids or the cells. And this is just part of the adaptation of Salmonella to the environment. We also identified several genes that have different functionalities for Salmonella in all the clades. Within them, we have 15 types of fimbria. And within these types of fimbria, some of them, the ones highlighted in red, had mutations. Mutations that can help us differentiate some isolates. For example, the CSGF and the CEF8 genes have mutations that were located only in the isolates within clade number 3. All the isolates lack the phage genes that belong to the reference PT4 strain. We also found SNPs that resulted in premature stop codons. These genes, due to these premature stop codons, are not functional. Within them, the CHBC that produces chitinases, the BCSA gene that participates in the synthesis of cellulose and is also associated to virulence. When the gene is mutated, the synthesis of cellulose reduces and virulence increases. And this gene is the SSEL gene, which is a virulence factor. If this gene is mutated, virulence is reduced. Regarding antimicrobial resistance, none of the antimicrobial resistance detected was associated with plasmids. We found resistance to 2 aminoglycosides, amikacin and tobramycin, in all the isolates from Mississippi. We also found resistance to ciprofloxacin and nalidixic acid, but only in the 2 isolates that belong to clade number 3. To contrast, this is the report from NARMS on the antimicrobial resistance of Salmonella enteritidis that's been isolated from human infections. In this situation, the resistance to ciprofloxacin and nalidixic acid are the most important, and they are increasing over the years, contrasting the findings of low level of resistance or a few isolates only showing resistance to these antibiotics in the Mississippi strains. Overall, Salmonella enteritidis from poultry in Mississippi are very similar and closely related to the PT13A US reference strain. We observe a reduced level of resistance to antibiotics, and it was not associated to plasmids. There is a negative selection for genes that confer antimicrobial resistance, and this might be the effect of all the effort the industry is putting in removing this antibiotic use from poultry. And the whole genome sequencing data from FSIS can be successfully used to identify possible targets for pathogen control. Within them, we can see the presence of the incompatibility X1 plasmid in some isolates, or the type 4 fimbria in the isolates, or also the isolates that belong to clade number 3, which are Salmonella enteritidis, are carrying resistance to ciprofloxacin and nalidixic acid. We have to also highlight the possibility of plasmid recombination events or co-integration of plasmids as an adaptation tool for the bacteria to better fit in their environment, to survive better in their environment and also in the host cells. So monitoring these events is very important in the detection of emergent and persistent Salmonella enteritidis strains in poultry. Our references. Thank you very much, and I look forward to your questions during the QA sessions. Thanks a lot.